So when you hear the word carbohydrate, what comes to mind? Probably things like bread or pasta, right? But in biology, carbohydrates are so much more than just a source of energy. They're the master architects of the cellular world. They're the structural engineers, and they're even the communication specialists. Today, we're going to dive deep into these incredible molecules, from their simplest building blocks all the way up to the giants that literally build life itself.
Here's the game plan. We'll start with the basics, then get into the really cool 3D shapes that give each sugar its unique identity. We'll see how they transform from straight chains into much more stable rings, how they link up to create huge polymers, and, most importantly, how that structure dictates their function. Then we'll wrap it all up with a review to make sure these key concepts really stick.
All right, so let's dive right in. What exactly are we talking about here? At its heart, the entire world of carbohydrates is built from simple repeating units that can be scaled up to some incredible levels of complexity.
We basically classify them by size. You've got your monosaccharides, those are the single bricks like glucose. If you link a few of those together, you get an oligosaccharide. And if you chain hundreds or even thousands, you get a polysaccharide. And their jobs are just as diverse. I mean, they're a main source of fuel, they provide the tough, rigid structure for things like plant cell walls, and they even act as little ID tags on the surface of our cells, which is absolutely critical for cell recognition and communication.
Now this is where it gets really fascinating. You see, a lot of different sugars share the exact same chemical formula. Same atoms, same numbers, but they are completely different molecules with totally different properties. So what gives? Well, the secret is in their three-dimensional arrangement, a concept we call stereochemistry. It's not just what atoms are there, but how they're arranged in space.
And the core idea here is something called chirality. A molecule is chiral if you can't perfectly stack it on top of its mirror image. The classic example? Your hands. I mean, go ahead and try it. Your left and right hands are mirror images, but there's no way to rotate them to make them superimpose perfectly. In molecules, this handedness comes from what we call an asymmetric carbon. That's a carbon atom that's bonded to 4 totally different things.
And a perfect example of this is glyceraldehyde. It's the simplest little aldose sugar. Here on the left, you see the L form, and on the right, the D form. They are perfect mirror images, but they are not the same molecule. We call this specific pair of non-superimposable mirror images enantiomers.
So, how do we know which is which? Well, there's a simple convention. In these flat 2D drawings called Fischer projections, we look for the asymmetric carbon that's farthest away from the carbonyl group up at the top. If it's hydroxyl group, the OH, is pointing to the right, we call it a D sugar. If it points left, it's an L sugar. And here's a wild fact. Pretty much all the sugars you find in nature are D sugars. Why? Because the enzymes in our bodies are also chiral, and they're built to recognize and work with only that D shape.
Okay, let's add another layer. What if two sugars are almost identical, but they differ in their 3D shape around just one single asymmetric carbon? Well, they're not mirror images, they're more like close relatives with one tiny difference. We have a special name for these: epimers.
This family tree of D-aldoses shows this perfectly. Okay, find D-glucose in the chart. Now, compare it to D-mannose right next to it. See? They're identical except for the direction of that hydroxyl group at carbon number 2. That makes them C2 epimers. Now, compare D-glucose to D-galactose. The only difference here is at carbon number 4, so they're C4 epimers. Just one tiny little flip creates a whole new molecule.
Now, while those straight-chain Fischer projections are great for understanding all these isomers, they don't really tell the whole story. Because in a solution, like, inside a cell, these sugars are constantly undergoing a really important transformation.
Here's D-glucose again in its open chain form. It's a useful drawing, but this linear shape is actually pretty unstable. In fact, at any given moment, less than 1% of the glucose molecules in a solution are actually in this straight chain form.
So, what's happening to the other 99%? Well, the answer comes from some basic organic chemistry. An aldehyde group and an alcohol group can react with each other to form a much more stable ring structure called a hemiacetal. And what's super convenient is that a monosaccharide, like glucose, has both an aldehyde and plenty of alcohols all in the same molecule.
So that linear chain just folds back on itself. The hydroxyl group on carbon 5 attacks the carbonyl carbon at C1, and this reaction snaps the chain shut into a stable 6-membered ring. But because the attack can happen from either above or below, it actually creates 2 slightly different ring structures. We call these anomers.
This ring-closing reaction creates a new and super important chiral center, the anomeric carbon. This is the carbon that used to be the carbonyl, and its new 3D configuration is what determines if the sugar is now in the alpha or the beta form. And believe me, that distinction changes everything.
And here's the simple rule to tell them apart when you're looking at these ring structures, which we call Haworth projections. If the hydroxyl on the anomeric carbon is pointing down, it's the alpha anomer. If it's pointing up, it's the beta anomer. That's it. Down is alpha, up is beta. Now, I know converting from the flat Fischer to the ring-shaped Haworth can seem a bit intimidating, so let's just walk through it. First, just imagine tipping that linear Fischer projection over 90 degrees to the right. Now, from there, a super simple rule applies. Anything that was on the right side of the original chain will now point down in the ring, and anything that was on the left will point up. And for all the D sugars we're talking about, that last C6 group always sticks up outside the ring.
Okay, so now we have our nice, stable monosaccharide rings. But how do we actually build those huge polysaccharides that are so important in biology? Well, the answer is a special kind of covalent bond.
This connection is called a glycosidic bond. It's formed when a hydroxyl group from one sugar molecule reacts with the anomeric carbon of another one. This is the fundamental linkage. It's how every single di, oligo, and polysaccharide in the natural world is built.
And this is where it all comes together. That little distinction we just made at the anomeric carbon, alpha versus beta, it might seem like a tiny detail, but the orientation of this one bond has massive world-shaping consequences. It perfectly illustrates that central theme of all biology: structure dictates function.
So, when you join glucose units with alpha-1,4 linkages, the chain naturally coils up into a loose, flexible helix. This is the structure of starch in plants and glycogen in animals, our main energy storage molecules. That helical shape gives enzymes plenty of places to grab on and start breaking it down, which allows for really rapid glucose release when your body needs a quick burst of energy.
But if you just flip that bond to a beta-1,4 linkage, everything changes. The geometry of that bond forces the polymer into a long, straight, incredibly rigid fiber. These fibers then line up next to each other and form strong hydrogen bonds, creating a structure with immense strength. And that is cellulose, the stuff that makes up plant cell walls. It's the exact same glucose monomer, but that one beta linkage turns it from a fuel source into one of nature's strongest building materials.
Okay, we have covered a lot of very specific terminology. So let's pull it all together in one place, just to make sure these relationships are totally clear for when you're studying.
Let's really organize this in a way that makes sense. We started with enantiomers, right? Those are the perfect, non-superimposable mirror images, like DNL glucose. Now, a broader category is diastereomers. That just means any stereoisomers that are not mirror images. So that means epimers, which differ at only one carbon, are actually just a specific type of diastereomer. And to take it one step further, anomers, like alpha and beta glucose, are a special type of epimer that only show up when this sugar forms a ring. You can see how these definitions all fit together. From a single hydroxyl group pointing left instead of right to a single bond pointing up instead of down, we've seen how tiny changes in geometry have massive consequences, turning a quick energy source into one of the strongest materials in the biological world. So the final question to think about is this: where else in biology does this fundamental rule that structure defines function change absolutely everything?